active DNA demethylation versus passive DNA demethylation. So in DNA demethylation, we learned that the 5-methyl cytosine is oxidized and it is converted to 5-hydroxymethyl cytosine. So what happens is the cytosine, okay, due to the addition of CH3 group, it gets converted to 5-methyl cytosine, and this reaction is catalyzed by DNMT, DNA methyltransferases, and this 5-methyl cytosine further can be oxidized in the presence of the substrate alpha ketoglutarate, which gets converted into succinate in the process. And this 5-methyl cytosine is converted to 5-hydroxymethyl cytosine due to the enzymes known as TET enzymes, 10-11 translocation enzyme. Okay, so now this 5-hydroxymethyl cytosine is further, it can be further converted to 5-formyl cytosine and 5-carboxy carboxycytosine, and those reactions are catalyzed by TET enzymes. Okay, and on the 5-formyl cytosine as well as 5-carboxyl cytosine, on both of them, an enzyme can work. Okay, sorry. An enzyme can work and it can convert it to the. Okay, it can convert it to the cytosine, and that enzyme is TDG, thymine DNA glycosylase, accompanied with base excision repair pathway. In the normal condition, 5-formyl cytosine and 5-carboxyl cytosine are the substrates of TDG. So this is what we have learned in DNA demethylation. This part, conversion of 5-methyl cytosine to the 5-hydroxymethyl cytosine or 5-formyl cytosine or 5-carboxyl cytosine, this part is DNA demethylation. This is demethylation of DNA. Okay, while conversion of cytosine to 5-methyl cytosine is DNA methylation. So we have learnt all of this till now. So today we'll be learning about what is the difference between active demethylation of DNA and passive demethylation of DNA. Okay, so the active demethylation of DNA is enzyme mediated. Means whenever the TET enzyme works on the 5-formyl or 5-hydroxymethyl cytosine, 5-formyl cytosine, or 5-carboxyl cytosine, all of these are the substrate of TET enzyme. Whenever TET enzymes works on this and it converts it into the cytosine, we call it as the active sorry, just a second. Whenever TET enzymes works on the 5-methyl cytosine and converts it into the 5-hydroxymethyl cytosine or 5-formyl cytosine or 5-carboxyl cytosine, this reaction is, if it is TET mediated, then we call it as active demethylation of DNA. While passive demethylation of DNA does not require any enzyme. Okay. So, you know, the active and passive, it can make a literal sense. Active means you are putting action, so you are working on it. Okay, and passive means it is just according to the flow. It is just within the, with time it's happening. That thing we call it as passive. You don't need to take an action to happen it. Imagine if you are writing something on board, okay, on the blackboard, you are writing something with a chalk, okay, and you there are two there are two situations. Either either you erase that part, whatever you have written with duster, or either you wait for a month, or either you wait and let it stay as it is, and after a month or after a couple of months, the, whatever written, the chalk writing, it will fade. Okay, it will fade out after as the time passes, it will fade. So here there are two situations. Here there are two kinds of the the chalk writing can fade in the two types. Either you do it by yourself, using duster. So this is the active active erasing of the chalk writing, and or you can wait and you can let it fade, and this is what you will call passive, passive fading of the chalk writing. So in the same way, DNA demethylation can be active or it can be passive. So in the active, it is done by enzyme, so it is tech mediated, just the way you are using duster, okay, for erasing the chalk writing. And passive is like you let it be, it does not require any enzyme. So what will be in the case of passing the time, in the case of cell, it will be division of cells. Okay, so whenever there is a rapid division of cells accompanied with inhibition of DNMT activity, DNMT1 activity. Why DNMT1? Okay, let's let's learn about it. So, when we learned about DNMTs, the members of DNMT family, we learned about DNMT1, which is a maintenance methyl transferase. We learned about DNMT3A and 3B, which are de novo methyl transferases. So DNMT1 acts on the DNA when you have to copy the methylation marks. Okay, when you have to copy certain methylation marks as it is. So what happens is that, see, this is the cell, okay, and this is the DNA. Okay, pardon my drawing. Okay, so this is DNA in the cell, and this is methylated. Okay, so this DNA is methylated on both of the strands. Okay, now what happens? That cell divides. Okay, cell has been divided, and it has now it has gotten into the two daughter cells. Okay, let's draw it in a prettier way. Okay, okay. So you have a cell and the DNA. Okay, it is methylated on both of the strands. Okay, so this is cell, this is parent cell. Okay, and now the cell divides. Okay, the cell divided and it formed two daughter nuclei. I am only drawing one. Okay, and when the whenever cell divides, the DNA, so replication of DNA is semi-conservative. Okay, so only now, so there will if one strand will be parent strand, and other strand will be the newly synthesized strand, so 50% hybrid, right? So one strand will be parent strand and one strand will be, so this one strand will be the newly synthesized. So parent strand will already have methylation marks, right? So this was the parent strand. So let's draw it in a different color. Okay, so this, no, not this, the newly synthesized strand, let's draw it in a different color. Okay, so this parent strand that already had the that already had methylation mark, while this pink one, this is newly synthesized strand. So newly synthesized strand won't have any methylation marks. So on this DNA, DNMT1 works, and it copies the methylation marks which was already existing in the cell, and it will convert this cell to a cell which is, it will convert this cell which is exact copy of its parent. Okay, now it will put the methylation marks on the both of strands. So this was parent strand already had a methylation mark, and now the newly synthesized strand will also have the methylation mark. So this is the work of DNMT1. But what happens if you do not let DNMT1 work? Okay, so in a scenario where DNMT1 will not work, it will be something like that. Okay, let's just copy this part. Okay, and yeah, so this, so when the parent cell will divide, it will form the daughter cell, and in the daughter cell, the newly synthesized strand would not have the methylation marks, and you are inhibiting the DNMT1 activity. So you are not letting DNMT1 act here. So what will happen? This daughter cell will again divide. Okay, so this daughter cell made two other daughter cells. Now both of the strands will be newly synthesized, right? So now those DNA would not have any methylation marks. So you are losing the methylation. So as the replication progresses, you are losing the methylation mark. What happens? Losing methylation marks. Okay, and this losing of methylation mark is known as passive demethylation. Okay, I hope now this is very clear to you all, that when there is rapid division of cells and inhibition of DNMT1 activity, that is what we call as passive DNA demethylation.